Perfect. So thank you everyone for joining. I'm very, very happy that so many people could be here today to actually celebrate our official kickoff of our HyLife-CETP project. So I want to very shortly introduce myself in the beginning. So my name is Nicole Dopfe. I'm the project coordinator of this project, and I'm joined here today by all our team members. And we want to give you a little bit of insight what our HyLife project is actually about, what we want to achieve, and how you can be part of HyLife also. So first, what will be discussed now today is I want to give you a little bit of a background. What is our CTP project? I already know that many of you are very knowledgeable on the topic, so I will keep it short, but still it's good to have some kind of background. And then the work package leaders will show some details and what the aims and goals of the technical work packages of our project. And then we will also present a little bit the research project partner so that you know who is actually working on this project. And then also in between, I want to take the time to show you that you can be part of HyLife and how this will work, and that we actually need a lot of strength and work together to make this a real success. And at the end, we will have time for questions. So because we are recording this session, there will be no questions in between. But we will have this dedicated Q&A session at the end. But just always put your questions, your ideas, comments, always just in the chat. They are not lost. And then we will address all the questions at the end, which will not be recorded. And the recording of the whole presentation will be afterwards available online for everyone. So with this, I want to shortly introduce you the background of our HyLife project, and this is hydrogen underground storage. I think most of you, as I said, are very knowledgeable on the topic, but just to explain it a little bit. So hopefully in the near future, we will have a substantial amount of our industry and society using renewable energy. But we know the problem with renewable energy, for example, wind or solar, is that it is not a continuous supply. So we have the problem, how do we store this renewable energy? And here hydrogen will be a very, very good option. And right now, really the only option we know when it comes to long-term storage. So what the idea is that excess energy, for example, when there's a lot of sun in summer, can be converted to hydrogen, and then this hydrogen can be stored. And then when there is energy needed, you can use the hydrogen again, either in direct use or you can transform it into power. So that is the idea. For these really, really large amounts of hydrogen, which we're talking about, there's actually the only option is underground storage. That means we want to store the hydrogen in the subsurface. And there are three options right now, which are salt caverns, saline aquifers, or depleted hydrocarbon reservoirs. And there's actually quite a lot of these gas storage sites already existing in Europe, and there's also a big potential to make more when it comes, for example, to salt caverns. But all the existing gas storage sites, they're dealing with natural gas, and as we know, natural gas is not hydrogen. And when it comes to the subsurface, also this subsurface is not sterile. So in the underground, you can find a lot of different microbes. Of course, not when you go to very high temperatures, but most of the reservoirs, all of the salt caverns, they are actually inhabited by microbes. And hydrogen for microbes is actually also a very, very interesting energy carrier. That means they can use the electrons from hydrogen, they consume it, and they grow on it. And this can actually cause a variety of different problems. For example, you can have H2S production, which is a very toxic and corrosive gas. Other microbes can produce methane or acetate, which means you lose your precious hydrogen. Microbes can cause reactions with minerals. They can also cause or influence corrosion. And if they grow a lot and build up a lot of biomass, they can actually reduce your injectivity and cause microbial plugging, which might be a problem for porous media storage. So a lot of things could be going on there, and there has been some research on it, but still it's not really clear how big the risk really is. And this is where our HyLife project now wants to start. So our main research question is this microbial presence, which is clear and a fact, is it really a risk for all the hydrogen storage sites? So what we want to find out to answer this question is that we want to sample a lot of different sites. And for this, first, we want to develop really some standards, some protocols, and some guidelines, how you approach a microbiology project when it comes to hydrogen underground storage. Because there's a lot of things already done, but everybody did it slightly differently. And now we really want to join together and do the same. And then we want to go out, sample a lot of sites, and then look at the differences. And this will give us actually the opportunity to find, or hopefully to find, the most favorable sites. Do we see some kind of indicators which are most important, where we then actually can say for future sites, these are the best ones? What we also want to do is that we want to look at if the microbial risks influence the technology and the business cases for these hydrogen storage sites. There are, of course, already business cases, but the microbial risks have not been really taken care yet, and there we want to really look from the microbiology standpoint on this topic. And at the end, the big question is really, we want to say, okay, how big is the microbial problem in all these salt caverns and the reservoirs in Europe? And how can we solve that? So these are a lot of big questions. So you can see here now all the partners who joined together for HyLife to try to solve this. So we all have a lot of experience in microbiology or related topics, and we want to sit together on one table and really define some protocols and some standards across all the partners to make all the data comparable. And with this, actually, with the advantage of all our knowledge, we want to go out and investigate as many storage sites as we can, and then also start a database on this type of data, really microbiology for underground hydrogen storage. And at the end, we hope to have really some guidelines, some tips at least we can give to the operators and governments when it comes to the microbial risks. So our big hope is that at the end of HyLife, we really can have aligned methods. We have the findings, which can also be then further used in other hydrogen storage projects. So this is now our work package structure. So our project will run for 3 years. And you can see now our 4 technical work packages. So as I said, first we have work package 2, which is the method alignment, which is very important because this will then funnel into the other work packages, especially work package 3, where we do the experimental testing and the bioinformatics. We will have work package 4 with a cross-cutting aspect where we look at mitigation and business risks. And work package 5 will summarize everything together in a bio-geomap and then also some guidelines which will come out of HyLife. So we will really start from a very, very detailed look at things or methods and microbes, and at the end have the large overview of the storage sites in Europe. So this is now the overall introduction of our project. And now I will give the word to the different work package leaders, which will a little explain their different work packages more in detail, what the goals and the aims are, and what you can expect out of it. So the first work package will be work package 2 with the method alignment. And here the work package leader is Katerina Czerna, and I will give the word to you.
So hello, welcome here at this kickoff meeting again. I would like to briefly introduce you the work package 2 that's called method alignment within the network and sample collection. I come from Technical University of Liberec, which is a technical university in the Czech Republic, in the northern part of our country. And the work package 2 is divided into tasks. And I will be now introducing mostly the task, the first task about aligned sampling, analysis, and the methodologies that needs to be developed first. And then Joachim will explain you more about the ways of sample collection and organization. So you can click to the next slide. So the first task of work package 2, and it's a very important one, is aligned sampling, analysis, and common methodologies. So we will start, or we have already started, discussing various sampling approaches, various methods how to analyze the data, and we need to align all these procedures among us so we can develop kind of a common protocol for all the partners. So if we analyze a sample, we shall all get to the same results. So we will be aligning geochemical methods, DNA-based methods, also the ways how to enrich and cultivate the samples, and very important will be also data interpretation. So we all follow the same guidelines. Once we will finish with this theoretical or mostly theoretical part, we will run interlaboratory comparison round-robin test, where we all share the same material, the same sample, and by the end of this testing, we shall all get the same data out of it. And this will help us to really prove that each of our labs can process the sample similar way. And lastly, the very important part of this task is continuous reassessment of best practices, because of course we will be learning by doing this project as well. So we will be developing and optimizing the protocols as well during the whole project. So we are sure that we will really work with the best procedure, or use the best procedures that we can use. And this whole alignment will be very important prerequisite for actual field sample collection as well as for the technical tasks. Next slide, and I give the speech to my colleague Joachim.
Hello, good afternoon. So I'm Joachim. I'm going to present you what we're going to do in this task 2.2. So our goal is to collect many samples from different storages, so in salt caverns and porous media in Europe, and to be able to compare the results we have. So for that, we need to standardize, to have some standards to in the sample collection, and so to be a little organization to be able to track our different samples and analysis. So for that, Katarina presented you our objectives on the sampling and methodology and alignment. So one of the first aspects of this task will be to talk, in fact, and to explain the different sample providers, the different storage operators, what is the methodology, what is the sampling methodology we retain, to help also those different storage operators during the sampling in particular. And the other part is to be able to track the different samples we have, and also to know in which laboratory will do the analysis, because we have 4 different microbial laboratories, and and so to track different samples, to have the necessary information to be able to compare the parameter of influence on the samples. So, for example, geological information, information on geochemistry, etc. And just to precise that we can the goal is to have a kind of pseudo-anonymized tracking of the sample, so the description of the sample providers. Okay, in short, it was a presentation of this subtask.
Okay, thank you very much. And with this, we go to work package 3, which is the assessment of the microbial impacts. And here the work package leader is Setare Rad.
Thank you, Nicole. I'm Setare Rad. I'm working in the French Geological Survey. So I'll be leading this work package, which is which is divided in 2 tasks. So the first task will be an experimental one, whereas the second one will be more about modeling and bio-IT. The person involved in this work package will be Sylvain Stéphane, who is working also at BRGM and I. And we have colleagues from Inria, Delphine Ropers, who will describe the task 3.2 after me, and Hide De Gong, who is also working in Inria, and we have a postdoc that just came in the project, whose name is Arnaud Belcourt. Next slide, please. So basically, the main purpose of the first task is really to have an approach field from the laboratory in order to mimic what is going on in different type of reservoir that we will have in the project. So for that, we will do experimentations, and we will characterize not only the microbial diversity, but we will also study the activities of the natural communities under hydrogen storage conditions. For that, we will have analysis for the initial reservoirs by geochemical data or DNA characterization. Of course, all this work package will start only when all the methodology will be aligned in work package 2 to be uniform in our field sampling method. And then we will follow by laboratory enrichments in different conditions. And also as they will, all those data sets will be input in a model in task 3.2. So we will discuss before with modelers to be sure to have very detailed studies with chosen hydrogen consumption conditions to have input in the model modelization part of the project. Next one, please. So I will briefly introduce the second part of this work package, which is based on modelization. To understand the aim of this part is more to understand the microbial kinetics, whereas it's by the physical chemical properties or geographically the geographical distribution of the microbial activities. And here are the details that will be explained by my colleague Delphine.
Okay, hi everyone. So indeed, in this task, our objective is to make sense of the geochemical and the DNA data by means of mathematical models. So concretely, we will map this data on the microbial community to metabolic functions using bioinformatic approaches that rely on the reconstruction of metabolic networks. So the idea is to identify the main metabolic functions that are involved into the consumption of hydrogen. Then we'll develop simple kinetic models that will describe the growth of the communities studied in the lab. And we will plug these simple models into larger 3D models that will describe those communities within the different sites. And the different models that we will develop will help us analyze these metabolic functions, understand the diversity of the communities and their metabolic capabilities across the different storage sites.
Okay, perfect. Thank you very much. And then we move on to work package 4, which is the innovative approach. And here the work package leader is my colleague, Annie Anstepich.
Yeah, hello. So our task or package 4 is the cross-cutting aspect of this whole project, which is called the innovative approach to hydrogen storage management. And our work package is aiming to develop an innovative and state-of-the-art microbial mitigation and management strategy by combining laboratory experimental results and with business analysis. So our work package is broken down into 2 main tasks. So task 4.1 is led by us at NORCE, where we will align methods across our microbiological labs and to investigate the efficacy of different inhibitors, both on the chemical and more on the green level, and to see how the hydrogen utilizing microbes respond to them under brine conditions. And the second task is led by Kain and her team from the Norwegian University for Science and Technology. And it will be also supported by Joachim from Geostock, where they will conduct an integrated economic analysis on understanding the value drivers of hydrogen storage facilities and assess the feasibility of various mitigation strategies. So overall, our work package will not only provide an overview on the efficacy of these common inhibitors, but also mainly provide a decision-making and economic analysis for subsequent sufficiency and sustainability evaluations. So if you can turn to the next slide. So our task 4.1 is broken down into 3 subtasks. And the first 2 subtasks are to compare and evaluate the efficacy of different biocides. And these will be funneled down into our third subtask, which is to define a blueprint of an optimal mitigation strategy, where we will correlate the different efficiency and efficacy of different biocides and evaluate their economic and ecological impacts. And all of these data will then be trickled down to the work package 5, where will be integrated into the biogeomapping. And in total, we will have 3 deliverables for this subtask. And all of these deliverables will also tie together with what will be generated for task 4.2. And with this, I'll pass the mic to Kain, who will describe task 4.2.
Hello, hi. This is Kain from NTNU. In this task, we will be working closely with a couple of colleagues from the NTNU as well as from Geostock. And we have broken it down into 7 subtasks, where the first subtask is to classify and categorize the storage facilities using the inputs from the earlier work packages. And then we are going to analyze or assess these external environments of the sites using the PESTER analysis, which is by looking at the political, economic, sociological, technological, legal, and environmental. And then afterwards, in the subtasks 3, we are going to look a bit more further to assess the environmental risk of a new underground H2 storage site using the ENVID method. And then we will look a bit closer into the internal intrinsic value of these categories of storage sites, or any mitigation strategies using the SWOT analysis, which is looking at the strength, the weaknesses, opportunities, and threats for each of these storage sites and the strategies. And based on the external and the internal analysis, we will then in the step 5 forecast cash flows associated with each category of the storage sites that we have categorized in the step 1. And finally, in stage 6, we are going to assess the total risks associated with each of these storage site category and the corresponding mitigation strategies that we have been talking about. And then finally, on stage 7, we are going to build an integrated economic model of storage facilities to support the business decision makings for the futures. So that is what we have for task 4.2.
Very good, thank you. And then we go to work package 5, which is the holistic data integration. And here we have our work package leader, Petra Bombach.
Yeah, good afternoon, everybody. My name is Petra Bombach, and I'm a microbiologist and work in the Isodetect company in Germany. I will lead the working package 5, which focus on the holistic data integration. So the working package harbors 2 different tasks. The first task is that we want to integrate all the hydrochemical, geophysical, and of course microbial data, and to summarize all of them in a huge database. And this task will be led by Jakub from the Technical University of Liberec. And the second task is that we want to derive general recommendations for actions for the industry based on the obtained results. So Jakub will now explain some details of the establishment of the database, and then I will explain more about the guideline.
Good afternoon, all. I'm Jakub from Technical University of Liberec, where I work as a bioinformatician, and I will be responsible for task 5.1, which will deal with design for database structure that will be suitable to store and to organize all the relevant data and the metadata. Which data will be included in the database will, of course, be a matter of discussion of all the project partners. Then we will summarize data on key geochemical and geophysical parameters such as salinity, temperature, and pressure, and of course microbial activity and composition of microbial composition into the database. And finally, we will set up a graphical user interface that will help the user to access all the data in the database, to export data, to visualize the data, to create maps, and so on. The deliverables will be the report on the design of the database, the graphical user interface, and publication on geochemical relationships. Thank you.
Yeah, as the outcome of the research project is of high relevance for the future underground hydrogen storage, we aim to derive general recommendations for actions. So we have the geophysical parameters, the hydrochemical conditions, the data on the microbial community composition, and of course from the lab experience under hydrogen atmosphere. And all these results are brought together in the task 5.1. And based on the obtained results, we want to propose good practice protocols and solution to ensure an efficient underground hydrogen storage with low microbial risk. And in addition, we want to determine the most important parameters from the microbiological point of view, and to summarize all these recommendations in a guideline for storage operators. Yeah, thank you.
Super, thank you very much. So these were now the presentations of all the work packages. I hope this gave you all a good overview of what we want to do. I'm very much looking forward to this project, because we can have actually quite a big impact on the hydrogen storage technology, because we really want to identify key indicators for the microbial risks. And that is very much needed, because if we succeed, we may be able to really pinpoint stable hydrogen storage sites in Europe, or at least provide some really good selection criteria, which will help to minimize the microbial risks for the storage site and also for the business. And we also want to develop the protocols. We want to have the reference data, which can be the baseline for future field trials also. And this can hopefully then enable safe, large scale and long-term hydrogen storage. And we also, by looking at really microbiological risk-specific business cases, hope to really drive this forward, so that the evaluation process is easier and more complete. And then also, because minimizing the risk, understanding the risk is one thing, but we also want, of course, then try to mitigate the risk. And here we want to look at mitigation options and management strategies. So at the end, we hope to achieve all this. So this was now the overview of our project. Before we now go to presenting more our research background and our research institutes, I just want to take already 1 minute to invite you all to actually be part of HyLife. Because you now understand maybe that we are actually need a lot of samples. We need a lot of samples to really gain the full picture we want to see. So if you are operating a potential storage site, you can be part of HyLife. At the end of the presentation, I will give you also again a little bit more detail, but very short. We will provide you with a sampling kit, the sampling procedure, and you then just send us your samples with some metadata, and we will analyze it. And then this will help us and will help you to put the data in the European big picture. This is, I think, a very, very good opportunity for us all to build knowledge. You will get an industry newsletter on the HyLife results, and you can be part of our network and hopefully a lot of good relations within the partners. But as I said, I will come back to this. Now we want to shortly present who we are as a research institutes. So every partner prepared 1 or 2 slides just to show our background and what we do and how we relate to HyLife. So I want to introduce NORCE. So NORCE is one of the largest independent research institutes in Norway. As you can see, we are located almost everywhere across Norway, and we are not only focusing on energy and technology, where Annie and me are located, but we are also very strong in health and society research and also environment and climate. Within the energy department, we have a very strong commitment to renewable energy, and there, of course, especially the hydrogen. And NORCE is actually leading 2 scientific centers on the project. You can see them here. So you can find them also online to check them out. They are also working a lot with hydrogen and some aspects of hydrogen storage. Annie and me, we are focusing on the geo and anaerobic microbiology part, focusing on industrial aspects. So with our background of sampling, cultivation, identification, and analytics. And we're also working a lot with high pressure and high temperature. And we now also have a dedicated lab to work with hydrogen under pressure. We are actually coordinating the HyLife project. And as you saw, we are also leading work package 4 with our background we have. Then the next one is Inria. So I would invite Delphine.
Yeah, so Inria is the French National Research Institute in Computer Science and Applied Mathematics. So here we work for the Inria Research Center at University of Grenoble in the French Alps. And so I'm leading a group of systems biology, which gathers microbiologists, computer scientists, mathematicians, physicists, so very interdisciplinary. And our common objective is the study of microbial growth, so that we analyze and engineer and control. And so, though we are working for an institute in computer science, our research problems are shaped by biological questions, and that we address by a combination of models and experiments. Next slide, please. And so those are examples of our research directions in the microcosm team. So for instance, on the top left, you can see examples of how we analyze omics data by means of bioinformatics approach that to analyze the metabolism of population of bacteria. And then with that, we are able to develop smaller kinetic model, coarse-grained models that allow us to study, like in the example on the bottom left, resource allocation strategies in microorganisms and at the population level, but also using some specific population models on the top right. We can analyze also how these research allocation strategies vary between the different bacteria in populations, and how those strategies are inherited from mother to daughter cells. And then we also study with models and experiments how a microbial community grow, and we try also to control them. And so in the HyLife project, we'll be responsible for the task 3.2. Thank you.
Then I give the word to Kain.
Yes, thank you. Again, this is Kain from Norwegian University of Science and Technology. It is an international-oriented university with headquarters in Trondheim. And we have campuses in Gjøvik and Ålesund. And the university's root goes back to 1760, but a merger in 2016 made the NTNU Norway's largest single university, with over 43,000 students and 8,000 employees, with almost 400 study programs. And the annual budget is of 10.5 billion Norwegian NOK. And we at the NTNU Business School, we are one of the main institutions for education and research in businesses and commercialization. So at the NTNU Business School, we have a Center for Banking and Finance, and also Center for Real Estate and Environmental Economics, where we have an international research network across 5 continents, and we are very active in various commercialization research, and as well as on the ESG, or the environmental, social, and governance issues. And we also have the Department of Mechanical and Industrial Engineering at the NTNU, who has a very extensive knowledge on the H2 related topics, and is a leader in the research area on safety and material integrity. So with that background, we are leading task 4.2 in the work package 4 in making this business case for the project. Yes, thank you.
Then we go to Geostock.
So as Geostock, so we are an engineering company specialized in the underground energy storage, and basically gases and hydrocarbons. And now we want to support our clients also with the new question we have in the frame of the energy transition. So we are providing services, can be consulting, engineering, construction of storage, and operation of storages. We have an expertise in different types of underground storage, being salt caverns, mine caverns, and porous media, such as depleted reservoirs and aquifers. And so we are involved also in developing new solution to for the carbon-free energies, so mainly hydrogen, and also we're working on CO2 geological storage. Here in HyLife, so we are leader of the subtask on the sample collection and management. We are also involved in geochemical and biological modeling. The graphic in the top right, so it's an example of a reservoir compositional model, including the reactivity, the biological reactivity for hydrogen, and we want to go further within this project. And so we are also involved in establishing economic models, and also on studies on evaluating the environmental impact of constructing a storage, so accounting for or not for to see the effect of the microbiological on the storages. Thank you.
Thank you. So, Sylvain?
Yeah, it's me.
Mm-hmm.
So, good afternoon, everyone. I'm Sylvain Stéphane, and I'm working with CETP at BRGM. So, BRGM is a French geological survey. So, it's a research institute that is working on all topics linked to subsurface and geoscience. So, you have in this slide some examples of the main topics where BRGM is involved. Quickly, we have some teams that are involved in geological characterizations in France, but also in the world. We have people that are working in the groundwater management, the geological risk, mineral resources, and the circular economy, the development of digital services and infrastructure. And of course, we are also working on topics linked to energy transition. So the HyLife topics is in this part. Next slide, please.
Mm-hmm.
Yeah.
No.
Yeah. CETP and I are we are a part of the microbiological teams of the BRGM. So we are a team of, at the moment, of 15 people that are working on different topics. So, for example, we have colleagues that are working on bioprocess, so for biolixiviation, bioremediation. We have also other colleagues that are working on the characterization of ecosystems and identify the function of microorganisms. So the idea is to determine bioindicators or to define the strategy of remediation. And we have also people that are studying specific geochemical processes. So, for example, in our case, it could be methanogenesis. We have, of course, several laboratories that allow to study microorganisms. So we have some pictures here. And we have also a high pressure platform that allows to reproduce deep environments that are characterized by high pressure and high temperature. So we call this platform BioRep, and we use and this platform is mainly used for energy transition topics. And we plan to use this platform to test microorganism properties in more representative way. The BRGM has worked since a long time on hydrogen topics, and the most recent project is called Ishiko project. And the aim of this project was to convert hydrogen and CO2 into a depleted reservoir to produce methane. So we will use our knowledge for the HyLife, even if the goal here is to avoid hydrogen consumption. So our main implication is in the work package 3, as CETP is leading this work package, but we have also small implication in other work package. Thank you.
Thank you.
Okay.
So,
yeah, sorry. So I will introduce briefly our university, which is technically University of Liberec. As I said before, it's in the Czech Republic, in the north, on the north from Prague. It's rather a small university, but we work in many different fields. And our research group is a part of Institute for Nanomaterials, Advanced Technologies and Innovations, which is a research center of the Technical University. And within this Institute for Nanomaterials, we have several research branches. And our branch is the Nanomaterials in Natural Sciences, where we focused mostly on research and development of advanced materials, mostly nanomaterials, and very important is that our focus is on environmental technology and protection. We focus very much on environmentally friendly and advanced technologies. And within this nanomaterials in natural sciences branch, we belong to the Department of Applied Biology, which is our department. And here we focus on several research topics. Me personally, I work in the field of geomicrobiology, and we deal with the microbial activity in relation to the future nuclear waste repositories. And now also we are interested in this hydrogen storage, which is new field to us. But also other colleagues work on different research topics, such as development of nanomaterials or various pearl structures for medicine applications, or we also focus on influence of nanomaterials and microplastics on natural microbial communities, and of course how it influences the natural ecosystems. So that's briefly from us. Thank you.
thank you. And then last but not least, Isodetect.
Yeah, Isodetect company was founded in 2004, and we are a spin-off company of the Helmholtz Center for Environmental Research and in Leipzig, and of the of the Helmholtz Center in Munich. So what we do, we provide microbial examinations and compound-specific stable isotope analysis to elucidate biological and also geochemical process in underground gas reservoirs and also at contaminated field sites, so soil and groundwater samples. And within the HyLife project, we will organize sampling at field sites, and we will be involved in the cultivation of the brine samples and reservoir samples under hydrogen atmosphere, and to determine the microbial hydrogen consuming processes by specific chemical analysis. And as I already said, we will lead the working package 4 dealing with holistic data integration and also data interpretation. Yes, thank you very much.
So thank you, everyone. So I hope you have now a very good overview about the project and who we are and what we do and what we want to achieve. So now I want to again take a few minutes and go into details how you actually can support our project. Because as you saw, and I already said several times, we always are looking for samples. And this is actually one of the big goals, that we get as many samples as we can. So how does it work? So you are now an operator of a storage site or a potential storage site, and you would be now interested to give samples, or maybe also you have samples anyway. Maybe there's some kind of work over planned, you will get samples, and then you say, we can also share some samples. So please contact us, let us know, and then let us know the type of storage and your preferred dates. So that means when can you sample and when would it be best for you. We can organize everything via email. We can also have, of course, meetings to discuss it a little bit more in detail as you like, and also how we plan it and about the data access. And then we will send you a sampling kit with a detailed sampling procedure and videos and everything you ever need to perform the sampling on site. And then you can send us the sample back to one of the partners plus the site information. And then this will be then funneled in our assessment. That means you will get then your analysis of your sample back, and you can also then be part of our network. That means this would really help us to see the overall trends in storage types in the different locations, the geologies, the chemistries. So this would be a really good opportunity for everyone to really get insight into the different microbial aspects. You can also be part of the advisory board. Then you can get invited to our workshops and our meetings and really follow HyLife as it goes along. So we are really hoping for you all to be part of HyLife so that we get the best possible outcome. Please contact us for this. And this is actually also now bringing me to the end. So I want to thank again everyone. We want to thank the CETP that they chose our project for funding and all the research institutions. I also want to give a big thanks to our industry partners, which made this project possible at all. So a big thanks to LANXESS, SLB, Corrosion Alliance, OMV, BP, and Total Energies. And I also want to give a big thanks to Stefan Jansen from Deltares, Julia de Rezende from Harriet Watts University, Anja Dormann and Martin Krüger from the BGR, because they helped us a lot in the pre-proposal, but unfortunately they were not eligible for funding. But they will still, their work is a lot appreciated. So, and this brings me to the final slide. Thank you everyone for joining our kickoff. Contact us in any case if you have questions. Follow us on LinkedIn. You will get regular updates there. We also have a website you can find also with this image. And I hope we could spark your interest in our project and that you help us to achieve the best possible outcome of it. And